Hello everyone! In the next minutes I will talk about our recent work, diffusion-based generation of histopathological whole slide images at the gigapixel scale. My name is Harb and I work together on this with Thomas Pock and Heimo Müller. Whole slide images are high-resolution digital scans of pathology slides containing, for example, human tissue or body fluids. These images are crucial in modern digital pathology and are used by medical experts for diagnostics, but also as input for algorithms in computational pathology. A special property of WSIs is their particular high resolution, reaching up multiple gigapixels. This is also related to the major challenge of our work. How can we sample extremely high resolution images using a diffusion-based approach? A major motivation to generate synthetic whole slide images is to alleviate the access to large WSI collections. All over the world, biobanks host large collections of tissue samples. Unfortunately, often privacy regulations restrict access to this valuable data for researchers. In contrast, synthetic images that retain pathological features of private training data, while being simultaneously unlinkable to individual patients, could be shared freely. Even though in the past many methods have been proposed that create synthetic data in histopathology, our work is the first deep learning based method that creates WSIs at the typically high resolutions. In particular, we create images with roughly 4 gigapixels. Since we cannot generate images of such high resolutions directly with a single model, we instead follow a coarse-to-fine sampling scheme where we stepwise increase the resolution. In particular, we first use a diffusion model to sample an initial low-resolution image. In further steps, we then add a relaxed super-resolution constraint to the diffusion model. This allows us to add finer details while at the same time preserving the coarse structure of images. Even though we do this patchwise at later steps, we avoid stitching artifacts by using a novel technique, grid shift. For our diffusion model, we rely on the previously introduced EDM formulation, where during training we have to learn a denoising function d parameterized by theta that is used to compute the score function. Contrarily to common implementations, we not only condition the denoising function d on the noise level sigma, but additionally also on the spatial resolution s of the currently processed image. Since WSI scanners usually save the spatial resolution in the metadata of scanned images, the spatial resolution is consistently available during training. Using this conditioning is crucial for our coarse-to-fine scheme. It allows us to set a high spatial resolution when sampling the initial image, such that a macroscopic overview of tissue is generated. Furthermore, in later steps, we then decrease the spatial resolution such that the model introduces finer details such as cellular structures in the image. To implement the denoiser function, we use a preconditioning where functions parameterized by sigma scale the network inputs, outputs, and a skip connection with the noisy input image. We train our model with score matching using patches of WSIs from our training data. Patches are sampled from random positions and uniformly across all spatial resolutions. After training our model, we then use our coarse-to-fine sampling scheme to generate high-resolution WSIs. In this scheme, we start from an initial low-resolution image and then sequentially upscale it in L steps. In each of these steps, the image from the previous step is split into patches that have the resolution of our model. Each of these patches is upsampled and the result is given by stitching the upsampled patches back together. To implement this upscaling, we guide the diffusion process with a super-resolution constraint, which is applied between the intermediate noisy images of the diffusion and the low-resolution patch from the previous image. Importantly, we do not strictly enforce the super-resolution constraint across all diffusion steps, but relax it by applying it only for the first R steps. Doing this relaxation is motivated by the fact that we face really high upscaling factors. For instance, if our model processes 512 sized images and we generate 65K sized WSIs, the overall upscaling factor is 128. If we would strictly enforce the super-resolution constraint across the full sampling process, even very small areas in the initial image would need to be consistent with large areas in the full resolution image. This would mean that even small errors in the initial image would influence large areas in the final image. This could be problematic. To implement the super resolution constraint, we replace the estimate of the fully denoised image at each step of the diffusion process with an updated one that is close to the original one, while additionally satisfying a super resolution constraint with the given low resolution image. We can express this with an optimization problem and obtain the solution using the method of Lagrangians. A major problem we face when using this patchwise upscaling is that without additional countermeasures, we would face stitching artifacts in the resulting images. A simple method to overcome this problem is to use overlapping patches and combine the super resolution with inpainting. This, however, comes with the drawback of increasing the overall runtime of our sampling while also not being trivial to parallelize. To overcome these problems, we introduce grid shift sampling instead. We interleave diffusion iterations with patching. After each diffusion step, we shift the patch grid that is used to extract lower resolution patches from the preceding image. This makes patch boundaries temporary, since they change after each diffusion step. Consequently, information between neighboring patches is continuously transferred, resulting in a more coherent result without visible seams. To train our model, we used 3,300 slides from the TCGA BRCA dataset. For sampling, we used a second-order Hoyne solver. In our setup, it took approximately 40 minutes to generate a single WSI. Looking at our results, one can see that our synthetic WSIs cover the whole magnification spectrum of real WSIs, starting from a macroscopic overview of tissue down to structures at the cellular level. To further evaluate the quality of our sampled images, we conducted a user study with professional pathologists. We presented them 20 synthetic and 20 real images and asked them to identify whether an image was real or synthetic. The results of our study suggest that pathologists were not able to consistently distinguish between real and synthetic images. So, to summarize our presentation, we presented a novel method to sample WSIs at unprecedented high resolutions. To this end, we use a coarse-to-fine sampling scheme where we gradually increase the resolution of an initial image. In this scheme, we guide the diffusion process with a relaxed super resolution constraint. Additionally, we introduced grid shift to avoid stitching artifacts. Finally, a user study with pathologists indicated that our synthetic images resemble the structure of real WSIs. Thank you for listening, and for more information, check out our paper.